all over the place. So there's two reasons I think that they're widespread in nature. One is they've got a very wide temperature growth range, including really most of them being very adept at growing at low temperature. Amazonensis is maybe the weird one, and there's a few that have been isolated from hot springs that are kind of on the upper side. The other reason that they're so widespread is that they have an enormous respiratory diversity. So the bacteria from the genus Shionella can breathe more compounds than any other organism that's been identified to date. And again, one of the reasons they're so easy to work with in the lab is that they prefer oxygen just like us. They can respire a whole host of other compounds that maybe some of those look familiar, maybe some of those don't. But then on top of all of this, they're able to respire a wide range of metals and metallic chelates and also oxide minerals. And what makes some of this very unusual is the ability to respire iron and manganese oxide minerals that at circumneutral pH in our aerobic world are minerals. And so these are images showing you on the left iron oxide, on the right manganese oxide, and just little bacteria that have been labeled with a protein called green fluorescent protein. And this just illustrates the problem here. So these bacteria eating food, inside their cell, they're breaking that food down into CO2 and protons and electrons. And those electrons, instead of reacting at the cytoplasmic membrane, which is where oxygen happens, this is what happens in our mitochondria as well, they have to deliver those electrons all the way to the outside of their cell. And so this is a process that has been, it's gone through a couple different names, but this one seems to have stuck, called extracellular electron transfer. So why do we care about this? From an environmental standpoint, the redox state of metals almost always influences the solubility or toxicity or both. Through the consequence of these bacteria respiring manganese and iron minerals, they dissolve those minerals. And so that influences the geochemical cycling of those metals. Respiration of insoluble substrates requires some unusual electron transfer pathways that I'll talk to you about. This trick also allows the bacteria to respire poised electrodes, and I'll explain that too. So we can take the minerals out of the equation and force them to make electricity for us. There are other researchers, so we study the fundamentals of the process. I have colleagues at the Naval Research Lab up in DC that are developing things called benthic microbial fuel cells that are run basically generating power from microbes in the sediment. Extracellular electron transfer may enable electrical communications between microbes in anaerobic environments. And when we get to that section, I'll kind of explain a little bit more about why that's interesting. So here are some of the different insoluble substrates. I showed you iron and manganese already. In these little bioreactors, this is a 3-electrode bioreactor where this little piece of graphite here is being poised at whatever redox potential we want. We can make it match manganese or make it match iron. The bacteria form a little biofilm on that surface. That's the only thing they have to breathe in this little reactor. And the way that they do this, so that for Shionella, they respire all of these substrates through the same pathway. The mechanisms get a little bit more nuanced, as I'll show you in a couple slides here. But the main pathway that they use is called the MTR pathway, stands for metal reduction. And basically, electrons from the inner membrane or the cytoplasmic membrane here are trafficked to the outside of the cell through primarily a series of multi-heme cytochromes. So all those red proteins there, multi-heme cytochromes, including MTRC on the outside, which is a decaheme cytochrome. This is a crystal structure of a paralog of that protein, but basically it's what MTRC looks like. MTRA is also a decaheme cytochrome. And so we understand this well enough that we can actually pull this entire pathway out of Shionella and make E. coli do this. My lab didn't do this, but Carolyn Aho Franklin's lab did this a couple years ago. So we really do understand this quite well in Shionella. So going back to the electrode system, this is the reaction that these little bacteria are catalyzing. They're eating lactic acid or lactate under anaerobic conditions. They're generating acetate. They get ATP from substrate level phosphorylation. And then they get these electrons that have to go away. And in these little bioreactors here, those electrons can be counted, every single one of them, as current over time. Okay, so that's part of the background on Shionella and extracellular electron transfer. And now I want to talk to you about some of the different mechanisms of how this works. Okay, so there's many different possible sort of ways to explain how this might happen. And so we did a perspective many years ago now on just kind of thinking about some of the different methods out there. So one idea is that the cell is directly in contact with that mineral surface. So the mineral is supposed to be the red block here. Iron 2 or reduced iron is the little red circle. Another idea that perhaps some of you guys have heard of in both the Shionella and Geobacter literature is this idea of nanowires, some appendages that are conductive in some way that allow electrons to move to that substrate. It's basically a riff on direct contact, but maybe would allow the cell to access something a little further away from itself. Chelators is another idea that certainly we know microbes out in the environment produce things like siderophores that are able to complex insoluble iron to make it soluble, and that's what's shown here. This works just fine for oxide minerals. This doesn't work for electrodes, by the way. And then this idea of electron shuttles, and this is what I'm going to focus on, where a compound that's produced by the bacteria and secreted acts as a proxy to deliver electrons to the insoluble substrate. Okay, so we know that Shionella can reduce substrates that it can't touch, which is one of the reasons why we think that electron shuttles is one of the main mechanisms for how this works. And this was done a couple of different ways. One was precipitating iron oxide in HPLC packing material. So this is a glass bead that has 50 nanometer pores that we precipitated iron oxide in. You can see a cross section of the cell. The cells outside of this glass bead were able to mediate reduction of iron all the way to the center of that glass bead. So that's consistent with a soluble compound helping mediate that reaction. Another group also did some work where they masked electrodes and showed that you got the same amount of current whether or not the cells could directly contact it or not. So there's some ideas out there that electron shuttling does occur in these bacteria. And what I'm going to do is walk you through some of the experiments that we did to basically come to this conclusion, at least under lab conditions. Direct reduction accounts for about 25% of this activity, and about 75% of this activity is mediated through electron shuttling. And we did this through the analysis of mutants that were unable to secrete this compound. Okay, I just told you that. All right, so we need a mutant that can't secrete. All right, so how do we know that Shionella produces electron shuttles, and what are they? I gave you a little bit of the background, but I'm going to tell you also how we stumbled into this. How are electron shuttles processed and secreted from Shionella? I'm going to tell you about 2 mutants that we identified in this process. And then finally, we'll look at our transport mutant unable to secrete this shuttle to come up with, to answer that question at the bottom. Are electron shuttles required to respire insoluble substrates? All right, so here's our little microbial fuel, or our little bioreactor again. Again, the bacteria on that electrode surface are generating current over time, and the electrons are derived from their ability to eat. So as long as there's food for them to eat in the system, they'll keep making current. But if they run out of current, if they run out of food, this current drops. So what we did, kind of through a serendipitous experiment, is we had a reactor at steady state, and we did a media swap. We basically just gave them a new source of fresh food. And when we did that, the current dropped. Even though there was a ton of food for them to eat, the current dropped almost 75%. And if we put the old stuff that we had removed back in directly, it restored almost all of the current to these cells. This was consistent with us removing something in the fresh medium that had accumulated in this older medium and that we had put back when we put the old medium back. And we did a bunch of experiments. This is some electrochemistry showing that over time, something accumulated in that culture supernatant that had a specific redox reactivity that was consistent with a flavin-like molecule. And then we went on to do some mass spec to actually determine that this is primarily a compound called flavin mononucleotide, or FMN. So what we see in young cultures is FMN, and older cultures, riboflavin. And this is what this molecule looks like. So this is vitamin B2, something that eukaryotes, it's essential for, but animals anyway don't make it. It's acquired from the microbes associated with them or from their diet. But most bacteria make this thing because it is an essential cofactor for all sorts of important redox reactions catalyzed by metabolism. So at that time, a colleague of mine in the UK published a very similar finding where instead of using electrodes like we were using, they were using iron oxides as the electron acceptor. We published a paper showing that flavins make electrode and iron oxide mineral reduction go faster. And also we showed that extracellular flavins are reduced by this pathway that I just showed you. But what we were really interested in is trying to identify a strain that doesn't secrete flavins. And to do that, we used a property of flavins, basically their fluorescence. So the way that we did this assay is we made random transposon mutants throughout the genome. And so this is a transposon that will, at random, insert itself somewhere inside the cell's genome and disrupt a particular gene. As long as that gene wasn't required for the cell's ability to grow under these conditions, we get a mutant that grows up. And we screen these mutants, kind of 96 at a time. We let them grow up, and then we simply looked for flavin fluorescence in the supernatants. And so we were looking for the rare mutants that would have less fluorescence than wild type, and then we tried to go back in and we would identify what was disrupted in these cells. And so over and over again, one of my early undergrads, Mike Torcha, kept hitting the same thing. And so we decided to kind of go after this. So let me tell you about this periplasmic nucleotidase. So we hit the same gene many times called USH-A. And here's a profile looking at intensity, fluorescence intensity over time. You see wild type has got this characteristic sharp increase in fluorescence compared to the USH-A mutant. So we see less fluorescent in the USH-A mutant strain. This is now a profile not looking at just bulk fluorescence, but rather quantifying FAD versus FMN versus riboflavin. And I'll show you what flavin adenine dinucleotide is in a second. So while wild type is in gray here, mostly we see FMN with a little riboflavin. This USH-A mutant had almost all FAD in it. And so this was quite unusual. So USH-A mutants in Shewanella accumulate less fluorescence in culture supernatants and shifts the flavin accumulation to FAD. So here's FAD, also an essential cofactor for a whole bunch of different processes inside of the cell. FAD is riboflavin, FMN is riboflavin with a phosphate on it, and FAD is FMN with an AMP molecule stuck to it. And when this moiety is attached to the flavin group here, it depresses the fluorescence. And so this molecule, FAD, has less inherent fluorescence than FMN. So we use that to set up an assay. We had hypothesized perhaps USH-A is involved in cleaving FAD into FMN and AMP. And if that were true, mutants that are defective in making this protein should lose this activity. So here's an activity assay where we're looking at FAD cleavage as judged by intensity in relative fluorescent units increasing. So wild type cells that have USH-A, they increase this fluorescence, while the USH-A mutant that we made is unable to do that, unable to cleave FAD, which is unusual. So this enzyme had been characterized for many years in E. coli. The E. coli enzyme can't do this at all. And we're collaborating with a structural biologist to try to figure out why that might be. But this is interesting. This is about some of the processing. This tells us that the flavins that are secreted by Shewanella is actually FAD, most likely, and that FAD gets processed by USH-A into FMN plus AMP. But this is not what we wanted. So what we did is we now started in an USH-A mutant background. So while wild type had a certain amount of fluorescence, USH-A had less. We now look for mutants that had even less. And this took us, so the same similar type of screen, to something called SO0702, predicted to be in this family of proteins called multi-drug and toxin efflux. So immediately this rang of something that the microbe might use to secrete something outside of the cell. And when we make mutations in this, and we called it BFE for bacterial FAD export, here's wild type making a certain concentration of flavin. So here in green is FMN, in yellow is total flavin. The BFE mutant makes just a tiny bit in the supernatant. If we put BFE in multi-copy, we can actually enhance the amount of flavins that come out of the wild type cell by increasing the abundance of that protein. So now we actually have a strain that we can do our experiments with. The BFE mutant still is able to biosynthesize flavins, but it just doesn't secrete them anymore. And that was important, and we've tried really hard to make flavin biosynthetic mutants, and we couldn't do it, which was also the story in E. coli. It's one of the reasons why, so flavin biosynthesis was one of the vitamin pathways that was most kind of recently worked out, because E. coli, flavins don't come back into E. coli, so you couldn't make a mutant. They ended up having to do a lot of that work in bacillus. Okay, so there's two different experiments here I'm showing you. Both are iron reduction experiments where we're looking at iron 3 being converted into iron 2 over time. And on the left is soluble iron 3 or chelated iron 3, iron citrate. And you see the mutants, the mutant and the wild type, and when we complement the mutant and the wild type, we all have the exact same reduction rates here. But when we use an insoluble substrate, we see a major defect. So here's the BFE deletion strain, reducing iron about 75% less than what wild type can do. And when we complement that by putting BFE on a plasmid, we see enhanced rates because we also saw enhanced amounts of flavins coming out of these cells. And we could do the similar experiment on electrodes. So here's our little bioreactor again. BFE mutants just make a small amount of current and don't really increase like MR1 does. So this is wild type here. And then if we take those same mutants, wild type and mutant, and just add a bunch more flavins to the bioreactor, we see that we recover the ability to produce current. And so going back to this model of what we think might be going on, so this is the MTR pathway of how electrons get outside of the cell, and an image of this crystal structure. What's interesting about this crystal structure, so you can see the 10 heme groups here. The 10 iron atoms are kind of the red balls here. Electrons enter this protein one way, but they go out of this protein probably 3 ways. And the idea is that perhaps out at the top is direct electron transfer, and the hemes 2 and 7 are within FMN binding domains that were only apparent from the 3D structure and really matched kind of what we had been seeing physiologically for a couple of years before Tom Clark did this work. Okay, so that's kind of where we're at in terms of the hardcore mechanisms of extracellular electron transfer. But you'll see why I wanted to give you that flavin background, because it will come up again in this next section here. All right, so now I'm going to introduce to you another microbe. So on the left is Shionella, the one I've just taught you about. This one is called Geobacter. Geobacter is a kind of a slightly different flavor bacteria. It also hates oxygen. It's an anaerobe. It's found primarily in sediments and in subsurface. But Geobacter also has a trick that Shionella never learned how to do, and that is Geobacter can stack on these electrodes. And because it can stack, a Geobacter culture makes a ton more current than a Shionella culture. And the cells way up here are still able to participate in electron transfer to that electrode through mechanisms by which a very few number of people argue vehemently about. Okay, so one of the reasons we wanted to put these things together is that I'm very curious about how microbial communities may have formed, and in this process, these two bacteria know how to do extracellular electron transfer. And in the anaerobic world, which again still exists in our guts, in the sediment right out there, and certainly in early Earth, the first billion or so years of life on our planet, the getting rid of electrons was really hard. In our aerobic world, oxygen is a great electron acceptor. It's really easy to get rid of electrons, so now we have different problems to life. But in anaerobic ecosystems, electrons disposal is a major problem. And so very curious about that. And maybe through engineering a community together, we can begin trying to understand those dynamics. Yes, and also just from a fundamental point of view, we've been studying Shionella for a long time on a plate or in a test tube or in a reactor, and yet in reality, it's almost certainly never alone. And so what could we learn about these organisms by studying it together with another one? And by putting one more organism in there, it gets probably more than twice as complicated, but certainly nowhere near as complicated as reality. Okay, so the system that we set up is basically, it's based on a carbon cascade, where we feed Shionella something, a substrate, glycerol. They make acetate as a byproduct of their anaerobic metabolism, which happens to be a substrate that makes Geobacter quite happy. So Shionella is an incomplete oxidizer of glycerol. Geobacter will completely oxidize acetate to CO2 by running a full TCA cycle. Now this looks good, except Shionella didn't know how to do any of this, so we had to take a bunch of parts out of E. coli, facilitator, a kinase, a dehydrogenase, and an isomerase out of E. coli to get this to work. So this shows Shionella growing, the engineered Shionella growing with glycerol as a carbon source versus wild type, which is unable to do it. We needed to do this because I needed an electron donor, a carbon source that we knew Geobacter couldn't use, and glycerol fit that bill. Okay, so here's the one basic aspect of this system, what we call a commensal co-culture. Feeding glycerol into the system, Shionella catalyzes that to acetate. Geobacter then takes that acetate to CO2, and they both know how to breathe this organic compound fumarate, making succinate as a byproduct. And so when we set those up, if we have just Geobacter alone, it doesn't know how to eat glycerol, and so we don't see any increase in optical density over time. If we do Shionella alone, we see Shionella growing pretty happily. Its growth is not dependent on Geobacter. But now in the co-culture, we see an increase in turbidity, and we can quantify that using colony forming units, showing Geobacter growing in the co-culture here versus when Shionella is absent, the inability of those cells to grow. We can also go in and do HPLC to confirm that acetate is actually transferring between these organisms. So if we look at Shionella alone, it's converting glycerol to acetate. We can see glycerol go away, acetate being produced at the expense of fumarate getting reduced. And then in the co-culture, we basically never detect acetate. As soon as acetate's made, it's gone. Okay, so that's an interesting system, but it's not super interesting. It's just a simple carbon cascade. And I mentioned to you, we're interested, can we force these bacteria to share electrons? And for that, we have another electrode system here. So now, instead of a graphite electrode, we switch over to a gold electrode. And for reasons that we don't really understand, Shionellas can't colonize gold electrodes. It's probably just a surface charge thing, but again, we don't quite understand that. But the idea in this system is Geobacter knows how to respire this electrode, but not eat the glycerol. Shionella doesn't know how to respire the electrode, but it knows how to eat that glycerol. But for every glycerol it converts to acetate, a bunch of electrons have to somehow go to that electrode because it's the only electron acceptor in the system. So let me show you that this does work. So here we're looking at current over time. So again, these are the electrons from the turnover of these carbon substrates. Geobacter alone, nothing happens because it doesn't know how to eat the glycerol. Shionella alone, it knows how to eat the glycerol, but it can't actually dispose of its electrons on the electrode. But in co-culture, we see current. And so we come up with some ideas about how the electrons might be moving between these cells. And there's some different ideas here. It could be that Shionella is making hydrogen. We know Geobacter likes to eat hydrogen. We know Shionella, in principle, can be making formate, although we never really see it in wild-type cultures. Maybe formate's moving electrons. Maybe it's using this pathway I just showed you, and there's direct electron transfer between the MTR pathway and some sort of reciprocal pathway on Geobacter. Maybe it's these flavin shuttles. So I'll show you, we don't know the answer to this fully, but I'll show you a couple of images here, or a couple of little bits of data. So here's a co-culture experiment. One of the first things that we did is we can make a strain of Shionella that's unable to make hydrogen anymore. So we can at least prevent the cell from directing electrons to make hydrogen. And when we do that, the system actually gets better. So that tells us that it's not interspecies hydrogen transfer. All right, so this is the mutant strain of Shionella along with Geobacter in this bioreactor. So then we tried this. We set it up with our Geobacter strain and our strain of Shionella, the BFE mutant that can't secrete flavins. And when that happens, we see that the system is impaired. And we can partially recover that activity by adding flavins back, which is consistent with the idea that at least to some extent, flavins are helping mediate interspecies electron transfer. I don't know if that happens in reality, but I'm very curious if that happens in reality. All right, so one of the things that we can do with this gold electrode is we can also go in and look at the spatial structure of the biofilm. And the images I'm going to show you are still super preliminary. Hopefully, if NASA funds this, we'll actually be able to do this a little bit more rigorously, a lot more rigorously. But one of the things I like about this is in most microbial ecology studies, you get a homogenized something. Everything gets mixed up, and you don't know who's next to who. But in this sort of system, one of the advantages of controlling it so well is we can go in and look at it. And in this Shionella strain that we've been using, we've got a GFP in the genome, and so we can immediately see where Shionella is. So this is the biofilm stained with DAPI, and so this is showing what is mostly Geobacter here. And then if we switch over to the green channel, we see where the Shionellas are, and what we see are these kind of little colonies of Shionellas, perhaps supporting larger colonies of Geobacter. There's another instance over here. And so this sort of spatial structure is something I'm quite interested in. And the dynamics of these sort of associations tells us something about the mechanism of electron transfer as well. If this required direct contact, the Geobacters around here probably shouldn't be working. Only the Geobacters in direct contact with the Shionella. And again, so this is still very preliminary, but it's an area that we're very interested in moving into. Okay, so flavin shuttles may mediate interspecies electron transfer in our engineered system. Maybe this happens in reality. Every single Shionella strain that we've ever tested secretes flavins, even ones that don't know how to do metal reduction. It's another reason why I've been very interested in this. And one of the things that we've also been thinking about is maybe moving into a system with even greater complexity, but still controllable. And that's something we call fluorescent in situ habitats, or FISH. So these are little zebrafish. This is a GFP labeled zebrafish that we use in our teaching labs, that is where the neutrophils are labeled with GFP. So it turns out, at least in we have two different zebrafish facilities at the University of Minnesota, one on the Minneapolis campus, one on the St. Paul campus. Both of them have got lots of Shionella in them, and they're not from my lab. That would have made things a lot easier. And so we've been playing the last couple of years with an isolate from the zebrafish. We call this one FH1. So this was isolated directly from the intestine of a zebrafish from the St. Paul campus. We know it's similar to a strain of Shionella called semiensis, which was isolated from China. The group that worked with that did a bunch in studying how these organisms can mediate dye decoloration of all things. The FH1 strain is similar to the MR1 strain in that they can both do iron reduction, or they have the ability to do extracellular electron transfer. They both secrete flavins. They both can respire fumarate. They also both really like TMAO. TMAO is a weird electron acceptor for Shionella because it's the only one I'm aware of that Shionella will breathe in parallel with oxygen. It just loves being in fish, especially if that fish is not alive. Okay, so we sequenced that genome. This was a combination of a little bit of nanopore sequencing with Illumina on top of it. One of the things that we noticed, so that most Shionellas have got a big megaplasmid that's got a lot of just kind of weird stuff on it. This one also had a small plasmid that contains things on it that might help the organism actually associate with the fish. So we're very curious about that. This is just doing a unique gene comparison. This is pretty common for most of the sequenced Shionellas, that there's a core, but there's always a bunch of stuff that's different. Okay, so one of the experiments that we can do really easily with zebrafish is make them azenic, and this makes them a very powerful system for microbiome experiments. So the work that I'm showing you here was done by a postdoc of mine along with an undergraduate. So what we do is we take these little embryos 1 day past fertilization. There's a kind of a regime of bleach and antibiotic and antifungal treatment, and let them kind of hatch in a sterile environment, inoculate them with a bunch of bacteria, and then euthanize them a couple days later and look at their guts and see how many colony forming units we see. So with just media only, we don't see any CFU of any microbes. Our wild type strain from upstate New York doesn't know how to colonize these fish, but the isolate from the zebrafish does. And so this is something I'm very curious about. We've got about a dozen Shionella isolates from zebrafish. All of them do metal reduction. Maybe that means that's how they kind of persist when they find themselves in a sediment and not in a fish. But that gut it becomes an anaerobic ecosystem, and maybe this process of electron transfer plays some role in their ability to colonize or persist in those fish. So I'm very curious about that. Certainly, it's not just about colonization, otherwise this guy would have done it, but maybe about persistence. All right, good. So in the last sort of 10 minutes or so, or maybe 8 minutes, I'm going to talk to you about this evolution story here. All right, so here is just a very basic thought experiment. And this is, just imagine a mixed environment where you've got one cell, this little green one, that grows really fast, twice as fast as these little black cells. And what happens over time in one of these mixed cultures or mixed communities is the fast-growing cell outcompetes that slow-growing cell in just a matter of generations, right? A couple of generations in, all of a sudden, you've got now twice as many of these green cells than you do the little black cells. But when your life depends on a surface, the dynamics we think might be different. And the idea here is perhaps fitness is uncoupled from growth rate. So the only cells capable of growth are actually the ones directly attached to our electrode. I'm not sure if this would translate into a gut, but I think it does translate to any surface. We might have a weird, we certainly do have a weird system. I mean, this electrochemical system is definitely not reality, but it's every cell attached to it, these are the only cells that can actually be actively growing, whereas the detached cells can't divide anymore. So the idea here is if we seed this electrode with a bunch of cells that grow really slowly versus a bunch of cells that grow fast, perhaps we can test this, that fitness may be changed when cells are dependent on a surface. Cell growth is dependent on a surface. And so for that, we have two strains. One is our wild type strain that has GFP in the chromosome. Another is a strain, it's a mutant that basically eats its food a little bit slowly. So it has a growth rate of about 60% compared to this strain. So this is our slow grower, this is our fast grower. And in just a mixed culture, so just a mixed culture tube, this is anaerobic conditions. If we start at about 5% fast cells, 95% slow cells, you see in just a few hours here, within a day, the fast-growing cells rapidly overtake that population. And we do this through flow cytometry, so we get really good numbers on this. You can't see the error bars. So that was in one of these test tubes. But what we wanted to do is actually test this in a bioreactor. And to do this, my student Eric had to kind of re-engineer the tops of these little bioreactors to basically turn them into a chemostat, a flow-through 3-electrode bioreactor. And so now this is the same experiment in this flow-through bioreactor where cells are actually respiring fumarate. So the electrode's not on, they're just in that reactor. And we see a similar thing. So we see the fast-growing cells rapidly overtaking the slow-growing cells. But now if we turn it on and let the electrode be the electron acceptor, and so note that this happens just in one day. We seed at 5% the fast grower, 95% the slow grower. We can go out for over a month and the population stay the same. And so what's really interesting about this, now again, it's an engineered system, but it must reflect some aspects of life depending on a surface out in the environment. I just don't know exactly what that is yet. But it seems to verify this idea that fitness can be uncoupled from growth rate if your life depends on a surface. So therefore, the dynamics of evolution could be different on a surface. And it's interesting to think about because most of our experimental evolution experiments are all done in planktonic mixed systems. And that's probably not really what life is always like. And so that's something we're quite interested in trying to understand a little bit more. All right, I talked very fast, so I'm going to acknowledge some people and hopefully have time for a beer and questions. So Annika did a bunch of this, the work on the co-culture. Eric did a bunch of the evolution work. My collaborator, Daniel Bond, where they're lighting methane on fire there from a little pond. And Peter has set up the zebrafish model system in our lab as well. And funding for the zebrafish stuff is still all bootstrapped on other things, but my main sources of funding are the ONR and NSF, and some internal funding from the U as well. So I would be happy to answer any questions if you have them. Yeah.
Well, I had a chuckle about your experiment here at the end because that's more of a real world situation.
I know.
Yeah. In the outside world, we're competing for space.
Yeah, I agree. And I think it's interesting. Maybe if you think about it, it's expected in some way. But our collaborator, who I guess I didn't acknowledge there, he's another professor at the U named Tony Dean, has about 9 pages of mathematical modeling that supports basically what we found, just based on theory. And so it's an interesting system to be able to actually test that theory. Yeah.
So we have some students in the audience here who are taking a course in geological oceanography, and one of the subjects we talked about was the anaerobic early Earth environment and the potential for origin of life in some different scenarios in terms of hydrothermal venting, open methane production. I was wondering if you could comment for the purpose of the class on some of your views on what you might consider might be a likely scenario. And the reason I ask this is because a lot of the work you're doing might be relevant for exploratory space travel, moons of Saturn, and so forth. We don't think about that a ton, but there are people that have written perspectives on how they think that iron respiration may have been the first form of respiration on the planet due to the abundance of the substrate. I'm not sure that there's good genetic evidence for that, but the main part of their argument is that if you go into these hydrothermal systems,
and you study these deeply branching archaea, a lot of them know how to do this. We don't understand the molecular mechanism like we do in Shionella and like we're starting to understand in Geobacter, but they have that metabolism. And so that, I think that's one argument for that type of metabolism on the early Earth.
So you said Shionella is a soil bacteria.
It's aquatic. It's a marine organism.
It is a marine.
Yeah.
Not in the soil.
You can find it all over the place, but often when you find them in terrestrial or freshwater systems, there is often some sort of marine connection. Not often. One of the model systems studied was isolated from putrefying butter, Shionella putrefaciens, right? Probably that was like in Iowa or something. But the genus itself was named after a researcher in the UK who studied low temperature spoilage of fish back in the 60s and 70s. And so that was more from a food microbiology perspective. But they can be, like my Google alert goes off whenever someone publishes something that has Shionella in the abstract, and half to maybe more than half are people just trying to culture things from fish that they're interested in, often marine, but not always marine.
Yeah. We have people studying bacteria and African dust that blows over here by the millions of tons, but I don't recall them ever mentioning Shionella.
No, they are not very desiccation resistant, but you know what they can do to that dust is they can make it soluble if they find themselves in the right environment. Yeah, definitely. Yeah.
Other bacteria doing that as well.
Interesting. Yeah.
Is the butter salted?
I don't know. Maybe that's actually a good question. We could go back and figure that out. There's a question in the back.
I'll ask the super simple naive question.
Good.
But, it essentially looks like you have like little bioreactors.
Yeah.
So is there any potential for those to be upscaled anyway?
Yeah.
And actually have some sort of industrial?
So that you also notice these were microamps. Like that sounds like a tiny amount of electricity because it is a tiny amount of electricity. People have installed these reactors. So when you in those particular systems, all the electrodes are in the same environment. So the electron kind of goes in and comes out and kind of reacts at the cathode right away. There's no barrier. But if you separate the cathode from the anode and put an ion permeable membrane in between them, you can force that electron to do work while it's outside of the circuit. So that's the premise for something called microbial fuel cells. And people have put them into wastewater treatment plants where there's a lot of food that nobody cares about. If you can convert some of that to electricity, that's nice. The more relevant applications seem to be in high carbon waste streams. I have a colleague of mine who installed one of these reactors in a Foster's brewery in Australia. And that brewery wastewater has got a ton of carbon in it, way more than what just normally comes into their wastewater treatment plant. And if you can use a system like that, the electricity is less interesting than just cleaning the carbon out of the water, making it less high strength. And then I mentioned too at the beginning, benthic microbial fuel cells, which could be independently run kind of low power sensors at the bottom of the ocean. The Navy has one right now in Pearl Harbor that's a magnetometer. So it has enough juice to be able to tell when ships are coming, and it matches like these logbooks almost perfectly from the people that actually run that stuff. So the Navy would ultimately like to be able to deploy these, and because they have sensors all over the ocean, as I'm sure you guys know, they have to put batteries in them, and it's incredibly expensive to do that. But still, we're talking low power applications. We're studying some other bacteria that run the opposite reaction. These are iron oxidizing bacteria that the neutrophilic ones, and the one that we study is called Mary profundus feroxidens from the Loihi seamount. It makes rust on the outside of its cell and brings the electron in. And because it's making something insoluble, it has to have a pathway to the outside world for just catalyzing the opposite reaction of Shionella. And we've grown those on cathodes, basically feeding them with electricity. And if we understood how that worked, we might be able to make bacteria feed them with electricity and make them make, say, a biofuel for us. And so like that's an area called electrofuels that is still a lot of theory, but people are making some progress in that too.
I'm sure you've heard about the president's proposed reduction in science by 20%. And I see that your primary funding sources are ONR and NSF, and I can understand ONR for the biofuel fuels. What are your thoughts about diversifying and looking into oil companies for alternative fuel sources?
Yeah, So one of my, actually the former postdoc that did some of this Mary Profundus work on cathodes is basically the head microbiologist at ExxonMobil. And I think this sort of stuff is still small scale for what they're interested in. They really want microbes that can do stuff to methane, like make methane into something more transportable, make methanol into something more valuable. That seems to be what, at least from the biotech side, what they're mostly interested in, although I haven't talked to her in a little while. She probably has too much money now. I don't know. I think when about 6 or 8 years ago, when gas was $3, $4 a gallon, like people talk, actually that strain that we built to eat glycerol, glycerol is a major waste product from the biodiesel industry. And the biodiesel industry basically doesn't, like it barely exists right now, but when gas prices go up, they'll come back and be competitive again. But they basically, they spray that on fields or they have to pay to dispose of it. And we thought maybe we can turn it into electricity for them. That didn't really pan out, but like we got some cool papers published and it ends up being the foundation of some of the new work in a fortuitous way. But yeah, when gas prices are low, I think these alternative types of energy methods are harder to sell.
So,
Yes, sir.
in the gut of the zebrafish,
Yeah.
do you find Show-Knowledge colonizing the mucus as a surface?
I don't know.
Oh, okay.
We are not nearly sophisticated enough. We can get the guts out and mash them up and put them on a plate, and that's about it. There's a group at University of Oregon that maybe knows more about that stuff, and they made some of the initial microbiome, at least the work that I'm most familiar with. They did some of the initial work on the microbiome there. Yeah, Gehrman. Yeah. Yeah.
Well, if there's no more questions, I think we're going to try to meet afterwards in a little while for TGIF. So you guys are going? Yeah. Continue the conversation there.
Okay, thanks.